Today I want to tell you a story of how we search for missing variants in large exome sequencing projects. And this is done by optimization of analytic pipelines. The data I present today are in application to Alzheimer's disease. So before I start, I want to first show you the agenda of my talk. In the first part, I want to introduce to you the rationale of our study, which can be summarized into 3 questions. What is genomic variant calling? Why is it important? And why does it need a petascale computer such as Blue Waters? In the second part, I want to tell you how we rescue additional variants by combining analytic pipelines. And here I will show you the big data workflow that's utilized in our study. And finally, I want to show you how we can apply our approach to the study of Alzheimer's disease. So going to the first part, what is genomic variant calling? Why is it important? And why does it need petascale computing? So most of you probably already know this, but a genomic variant is a difference in a genetic code. So say, for example, we have an original sequence here on the top, and this person has TAA-CTG. But in a patient, instead, we have TAA-CCG. So at position number 5, we have a C instead of a T. This C is a variant. And genomic variant calling is identifying variants from the genome. So why is variant calling important? It is important because genetic variants are known to be associated with the disease, with the risk of many diseases. One famous example is the ApoE variants in Alzheimer's disease. The E4 allele can increase the risk of getting AD by 12-fold. And the E2 allele, which is more rare, can actually decrease the risk of AD by up to 40%. And this is certainly not the only example here. Nowadays, variant discovery by next-generation sequencing has become a common method for identifying disease mechanisms. So lastly, why does variant calling need petascale computing? This is because nowadays sequencing projects are becoming more and more large-scale. For example, Centers for Common Disease Genomics plan to sequence about 200,000 whole human genomes. And one sample roughly equals about 200 gigabytes of sequencing data. So 200,000 samples would roughly equal 40 petabytes of sequencing data. And that is the input data for the variant calling practice. So now you can see that this would require both large storage and fast processing. And this brings up the question. The current variant calling practice were established about 10 years ago based on small sample sizes. But is it still sufficient in today's large-scale projects? And that is a question our study tries to answer. And here I want to tell you of our research, rescuing additional variants by combining analytic pipelines. So let's first reveal the current default variant calling workflow. And this can be summarized into 4 stages. The first step is sequence alignment. In another word, at this stage, we align the sequence of our samples to the human reference. The recommended tool for alignment is BWMM. Following alignment, the next step is multi-sample genotyping. At this stage, we identify mutations from our samples based on evidence across all samples. Following that, we do variant filtering to remove low-quality variants. And after that, we end up with a list of usable, high-quality variants. So from this workflow, you can see that it typically uses 1 aligner and 1 genotyping strategy. So the question is this still enough when we have thousands of samples? To answer that question, we used up to 10,000 exome samples to test the 2 main steps in the workflow. The first test we did is aligner comparison. So in addition to use the default BWMM aligner, we also use a NovoAlign, which is another popular alignment tool. This is important because different aligners tend to prefer different types of variants or variants in different genomic regions. So in large sample size, the collective differences caused by aligners may be big. And if that is true, that could lead to lots of missed variants. To address this question, we tested 6 different sample sizes, starting from 50 to 1952. And these samples were selected for an early onset AD study, which I will talk a little more later on. So here's our results. At sample size of 50, you can see that about 91% of variants are identified by both aligners. Now this percentage decreases as the sample size goes bigger. At our final sample size of 1952, we see that only 73% of variants are identified by both aligners. To translate that percentage into numbers, that means about 63,000 variants are only identified by using BWMM, and about 42,000 variants are only identified by using novel align. So if we use the default approach of one aligner, these are the variants we will never see. So the next obvious question is, do we care about these variants? Are they good quality, and are they important? The short answer to these questions is yes, and I'm going to show you that in the next few slides. So to learn about the quality of our rescued variants, we looked into several main variant quality metrics. The first thing we looked at is B allele proportion, which is the percent of reads that supports the alternative allele. And we compare that across 3 groups of variants uniquely identified by BWA, by novel align, and variants identified by both aligners. And here we see that 3 groups of variants have very similar distribution. You see a peak at around 0.5 corresponding to heterozygous, and a peak at about 1 corresponding to homozygous alternative. This is what's expected out of a diploid genome, such as a human genome. The next thing we looked at is sequencing quality, or GQ score. And again, you can see all 3 groups have very similar distribution, and they peak at 99, which is the highest value we can obtain. The third quality matrix we'll look into is sequencing coverage. And again, you can see that all 3 groups have very similar distribution of high depth. And lastly, population frequency. 3 groups of variants have very similar distribution, but the 2 aligner-specific variants actually have a higher percentage that are more rare in the population. We further looked into additional characteristics in the 3 groups of variants. We see that the 3 groups have very similar chromosome distribution. The majority of them are located in reasonable genomic regions with no mapping difficulty. The 3 groups of variants have very similar GC content surrounding them. They have very similar functional impact separation. And what's interesting here is that the more of the aligner-specific variants are actually novel, which are the very type of variants we're interested in. So to conclude from our aligner comparison test, we see that using an additional aligner rescued a significant percentage of good genetic variants. The impact of rescue is correlated with sample size. With bigger sample size, we get higher percent of rescued variants. Now moving on to our second test, which is genotyping strategy comparison. So here, in addition to multi-sample genotyping, we also performed single-sample genotyping, which finds mutations based on the sample and the sample alone. This is important because multi-sample genotyping is based on evidence across all samples. So if we have a variant that's only present in one sample or a few samples, the evidence may seem inadequate in a scale of large sample size, and this type of variance could be missed. To address this question, we also tested 6 different sample sizes, starting from 50 and ending at 10,000. And here's our result. At sample size of 50, single-sample genotyping added about 10% of additional variance. And this percentage goes up as sample size increases. At our final sample size of 10,000, we see that single-sample genotyping added more than half of total variants identified. To translate that into numbers, that is 0.8 million variants that we will never see if we only use the default pipeline. Similarly, we also looked into the quality metrics of the rescued variants, including percentage of supporting reads, sequencing quality, sequencing coverage, and population frequency. And here we see that the variants rescued by single sample genotyping have good quality, and more of them are rare in the population. Similarly, the rescued variants and the shared variants also have comparable characteristics in terms of chromosome distribution, genomic region, GC content, functional tiers, and in addition, more of the rescued variants are novel. So to conclude from our genotyping strategy comparison, we see that using an alternative genotyping strategy rescued a significant amount of good genetic variants. The impact of rescue is correlated with sample size. With bigger sample size, we get higher percentage of rescued variants. Now for the next minute or two, I want to show you how we can apply our approach to the study of Alzheimer's disease. From the 1952 samples that we mentioned earlier, we actually combined both aligners and both genotyping strategies. And we wanted to see how many additional variants we can find in 3 known early onset AD genes, including APP, PNS1, and PNS2. As it turns out, 4 out of 13, or 31%, of previously published variants were missed by the default pipeline, and they were rescued by our combined approach. This is important because this both validates our approach, but also alarms us that using the default pipeline could lead to many missed opportunities of uncovering disease mechanisms. To summarize our study, I hope that by now I've shown you that using multiple analytic pipelines rescued a significant amount of good genetic variants. Large sequencing projects particularly need a complete variant call sets from multiple read to variant pipelines. We are currently testing our observations further on blue waters. This includes larger sample size, more extensive list of tools and parameters, and estimating the computational resources necessary. The project is currently ongoing. To a recent Blue Waters Award, we will carry this project further. Finally, I want to acknowledge the people involved in this study. This project is a collaboration between Mayo Clinic and UIUC and CSA. At Mayo Clinic, this work was led by Dr. Yan Asman from the Bioinformatics Department, with collaborations with PIs from the Neuroscience and Biostats Department. At UIUC, this work was led by Dr. Luda Minzer, with great help from Dr. Matthew Hudson. Our manuscript on this topic was recently published, so here's the information if you want to read it further. With that, I would like to thank you for your attention.